Hi everyone, welcome to another neurolinguistics paper. Lately I've been getting very interested in how bodily hormones affect the development of brain areas associated with language. On this topic, today we'll be talking about a paper from 2015 called Sex Hormones in Early Infancy Seem to Predict Aspects of Later Language Development. Let's first jump in with some background. They open the paper with a quick review on what we know from songbirds about sex hormones and linguistic functions. Songbirds, especially a species of zebrafinch called T. guttata, are popular models for vocal learning in animals. Using songbirds, scientists have discovered that estrogens within the brain, called neuroestrogens, seem to play an important role for songbirds in the ability to learn song. These neuroestrogens seem to affect the development of auditory circuits as well as their auditory learning process. These studies on vocal learning are the closest that we can come to language studies in animals, since language is an ability unique to humans. In terms of direct human studies of hormones in language, some have related prenatal hormone levels to later language performance. For example, one study showed that fetal testosterone levels negatively predicted vocabulary development at 18 months and 2 years of age. And similarly, cord blood testosterone levels in baby boys was shown to negatively predict vocabulary at 2 years. Let's move on to how sex hormones have been shown to affect the brain. Animal evidence has shown that a main type of estrogen, called estradiol, has been known to affect the neural organization of regions such as the amygdala and the hypothalamus. There seems to be more evidence for how testosterone affects the brain, including a human study showing that testosterone affects cortical thickness throughout development. Testosterone is also thought to influence cortical asymmetry and lateralization differences between boys and girls, and has even been shown to affect adult brain structure after less than 2 months of hormone treatments in a cohort of adults transitioning from male to female. So basically, there's quite a lot of evidence that the sex hormones estradiol and testosterone affect brain structure and development. This brings us to a discussion of one of the main waves of hormonal signals in human development. Most everyone has heard of puberty, which happens around the age of 13 for most kids and induces lots of changes associated with reproductive, bodily, and mental maturity. But fewer people have heard of mini-puberty, which is a postnatal period where sex hormone levels rise. This period occurs between 3 and 6 months of age, but its exact role and mechanisms are not quite certain. It's thought that it creates the basis for future fertility and allows the masculinization of genitalia in boys, but how this exactly happens and its role in girls is still quite mysterious. The figure on the right shows hormone levels in the solid line in both pre- and post-gestation time points. The top graph shows the fluctuation of testosterone levels in baby boys, while the bottom shows estradiol fluctuation in baby girls. Note how both peak around 2 to 4 months of age and then gradually drop off at around 5 or 6 months. The present study took hormone measurements at 5 months of age, shown by the red vertical line. The effects of mini-puberty on language have been somewhat studied in neonates, and it's been shown that testosterone level is linked to phonological discrimination ability in newborns, while estradiol is positively related to infant cry melody. So considering these previous publications, the authors come up with this hypothesis. They say the serum concentration of testosterone and estradiol during postnatal sexual hormone surge influence the neural substrate relevant for language behavior. Seems legit. Let's look at their experiment. Their participants consisted of 20 babies at 5 months of age, 9 girls and 11 boys. At 5 months, the researchers took hormone measurements of testosterone, estradiol, and a protein called sex hormone binding globulin, or SHBG. Then, approximately 3.5 years later, when the participants were 4 years of age, they returned to perform a language task called the SETK, which is a German acronym that translates to the German Language Test for 3 to 5-year-old children. This test contains 5 subtests, which are supposed to measure different aspects of linguistics, and we'll look at this list in the next slide. Now, the novelty of this study is that before this, no studies looked at the effects of hormone levels during mini-puberty on behavior at such a later age. So the researchers then took a look at the kids' results on the language measures at 4 years of age. The 5 language measures consisted of sentence comprehension, morphological rule constitution, sentence memory, memory span for word order, and phonological working memory. They found that between the boys and the girls, there was no significant difference between their scores on these measures. There was also no significant impact from variations in age. The researchers then used a regression model to look at the relationship between hormone levels at 5 months of age and scores on the 5 language subtests. They found no significant association between hormone levels and sentence memory, memory span for word order, phonological working memory, and morphological rule constitution. It's also worth noting that 3 of these skills are directly memory related. However, they did find a significant association between hormone levels and the sentence comprehension subtest. They found that testosterone was negatively associated with performance on this language measure, whereas estradiol level was positively associated. That is, higher testosterone levels in both girls and boys at age 5 months is associated with lower sentence comprehension scores at age 4 years, with the opposite being true for estradiol. Now, in this figure of the paper, they report the blood concentration, aka the serum level, in the top row. In the bottom row, they report what's called the free androgen index and the free estradiol index, plotted against sentence comprehension score. These free hormone indices are another way to report hormone levels, and they're calculated by dividing the serum level by the level of sex hormone binding globulin, which, if you remember, was also measured at the 5-month time point. This protein binds free-floating sex hormones, so the free indices are essentially a measure of how much hormone is left free-floating and active in the blood. Overall, the results shown here indicate that sex hormone measured at 5 months differentially impact sentence comprehension at 4 years. Next, the researchers calculated what they call the testosterone over estradiol quotient by dividing the serum concentration of testosterone by that of estradiol. They found that this quotient was negatively related to sentence comprehension, and we can interpret this to mean that a larger difference between testosterone and estradiol levels predicts worse sentence comprehension in both boys and girls. So where does the brain come in? Conveniently, the authors happened to have electroencephalography, or EEG, data on these kids while they performed a language discrimination task at 5 months of age. Apparently, this was data that had been published in an earlier study on the same cohort of kids. The language discrimination task involved something called a passive oddball paradigm. This paradigm involves presenting the participants with a standard phoneme, such as ba, which occurs frequently, and a deviant phoneme, such as the longer ba, which occurs less frequently. The idea is that if the child is able to tell the difference between the two phonemes, their EEG activity will reflect this in the form of something called a mismatch negative. The mismatch negative is part of the trace collected during EEG and happens when an unexpected change occurs in an otherwise predictable sequence of sounds. Therefore, it can be used to test phonological discrimination in infants. The researchers found that these mismatch negatives collected at 5 months negatively predicted sentence comprehension at 4 years. Unpacking this is a little counterintuitive. Basically, a more mismatch negative amplitude indicates a larger brain response to the deviant phoneme, which indicates better phonological discrimination. Therefore, we can say that better phonological discrimination at 5 months was associated with better sentence comprehension at 4 years. The authors then constructed a new regression model using the mismatch negatives, testosterone levels, and estradiol levels at 5 months. By doing this, they found that these 3 variables could account for 63% of the variance in the sentence comprehension test. Mismatch negatives were negatively related, which, as we've said, indicates that better phonological discrimination predicts better sentence comprehension, while testosterone was significantly negatively related to sentence comprehension, and estradiol was positively related to sentence comprehension and was also found to interact with the mismatch negatives. So what does this mean in the context of past research? For one, these results definitely corroborate past findings about prenatal and infant hormones, supporting that testosterone may negatively impact language skills, while estradiol may positively impact language skills. These results also bring up the possibility that sex hormone levels could potentially be used to predict later language ability, which could be useful for therapeutic interventions. And very interestingly, the results show that hormone concentration and brain activity in the form of mismatched negatives from EEG can interact to affect language comprehension later in life. Now, this study doesn't involve any sort of spatially precise brain imaging, so we can't relate the hormonal and behavioral data to specific regions of the brain that may mediate these linguistic processes. However, the researchers bring up that the inferior frontal gyrus may be playing an important role here because of some previous research. This study from 2010 found that in a young adult cohort, there was a negative association between testosterone and IFG gray matter, while estradiol showed a positive association with gray matter in this area. This is all good and moderately interesting, but why should we care about this brain area in particular? Well, this particular brain area is also known as Broca's area, and has been known to be involved in grammatical processing since the 1800s. Moreover, this region is known to undergo dramatic changes in gray matter volume in preschool and school-aged children, and these changes are actually correlated with comprehension performance in complex sentences. And yet more research has implicated this region in sentence comprehension in both adults and 6-year-olds. To conclude, this paper showed that testosterone and estradiol levels at 5 months of age affect language comprehension at 4 years of age, and that this could be an important factor in neurolinguistic development. However, more work is necessary to discover exactly how different sex hormones affect individual brain regions at specific points in development. Thank you guys for watching. I hope this presentation was helpful and educational, and if you'd like to see more neuroscience linguistics content, then go ahead and click the subscribe button below. See you guys next time.